Good morning, everyone. Today I'm again welcoming you with a new topic with the application of genetic engineering in plants. So, as we know that agriculture, or more specifically, the cultivation of plants, is the world's oldest biotechnology with an unbroken history that strikes back at least 10,000 years. Throughout this period, humans have constantly searched for improved varieties of the crop plants. Varieties with the better nutritional quality, higher yield, or features that aid cultivation and harvesting. During the first few millennia, crop improvement occurred in a sporadic fashion, but in the recent centuries, many varieties have been obtained by breeding, programming of ever-increasing sophistication. However, the most sophisticated breeding program still remains an element of chance dependent, as it is on the random merging of parent characteristics in the hybrid offspring that are produced. The development of a new variety of crop plant displaying a precise combination of desired characteristics is a lengthy and difficult process. Gene cloning provides a new dimension to the crop breeding by enabling direct changes to by the made of the genotype of plant, circumventing and the random process inherited in conventional breeding. There are the two strategies have been used for this. One is gene addition and another is the gene subtraction. So in gene addition, the cloning is used to alter the characteristic of plant by providing it with one or more new genes. While in the subtraction, the genetic engineering techniques are used to inactivate one or more of the plant's existing genes. So now we come to our topic. Here we will discuss about pest and disease resistance, abiotic stress tolerance, and production of some useful products produced by this genetic engineering from the plant. So this is a list of some successful genetic engineering projects. A number of projects are being carried out worldwide, many by biotechnology companies, aimed at exploring the potential of gene addition and gene subtraction in crop improvement. Here we will see that the GM crops, the genetic modified crops that synthesize delta endotoxin or glycophosphate resistance enzyme, are by no means the only example of plant engineering by gene addition. I am talking that the delta endotoxin, which is Bacillus thuringiensis, it's used for BT cotton, BT tomato, and now it's some BT brinjal also. So this actually the list of some additional projects which are completed already. So these projects included an alternative means of conferring insect resistance using genes encoding for proteinase inhibitor, small polypeptides that disturb the activities of enzyme in their insect gut, thus preventing or slowing the growth. Proteinase inhibitors are produced naturally by several types of plants, notably legumes such as cowpeas and commonly beans, and their genes have been successfully transferred to the other crops, which do not normally make significant amount of these proteins. The inhibitors are particularly effective against beetle larvae that feed on seeds, and so may be the better alternative than this delta endotoxin for plants whose seeds are stored for long periods. Other projects are exploring the use of genetic modification to improve the nutritional quality of crop plants. The beta carotene, that is vitamin A, contents of cassava has been increased 20-fold by the addition of genes for phytotin synthesis from the bacteria Pontia, or D-xylulose 5-phosphate synthesis from Arabidopsis thaliana. The iron and the protein contents of cassava has been improved, the former by the introducing a metal transporter protein, chlamydomonas, and the later with the protein called porogene. This gene is a fusion between the sporin, a protein from sweet potato tubers, and zinc and from maize kernels, designed to combine the best nutritional quality of the sweet potato and maize protein. Cassava is a stable crop in many parts of sub-Saharan Africa, but dependency on unmodified cassava leads to vitamin A and iron deficiency, especially among the children. The development of a bio-fortified version of cassava could therefore have a significant impact on nutritional and health part of Africa. In an analog project, biofortified maize with improved beta-carotene, ascorbate for vitamin C, and folate for vitamin B9 contents has been engineered by transfer of genes from rice and the bacteria Escherichia coli and Pontia. Finally, in the different sphere of commercial activity, ornamental plants and unusual flower color are being produced by transferring genes for enzymes involved in pigment production from one species to the another. So, up to here we have seen a little introduction about what is the genetic engineering, why is genetic engineering used for the improvement of plants. Now we coming to our theme, that is how to improve that agronomical traits by the genetic modification. So as we have seen that for centuries plant and domestic animals have been bred to select most desirable traits, either in terms of their appearance, hardiness, or yield of useful products. In conventional breeding, the farmer or breeder rely on chance recombination events to bring together beneficial combination of genes when particular processes are performed. Genetic engineering provides a shortcut to conventional breeding by introducing such genes directly into the genome. Genetic engineering also removes the constraint of sexual compatibility when carrying out process. Since genes from any source, bacteria, animal and plants, can be transferred into the recipient organism. Nowhere are benefits of genetic engineering or traits improvement more apparent than plant biotechnology, given that commercial genetically modified crops now cover over 75 million hectares of farmland in over 20 countries. Now we discuss some of the agronomical traits in plants that have been improved by the genetic manipulation. So first we discuss about the herbicides. Okay. Herbicides are useful to kill weeds and generally affected processes that are unique to plants, such as photosynthesis or amino acid biosynthesis. Both crops and weeds share these processes, and developing herbicides that are selective for weeds is very difficult. An alternative approach is to modify crop plants so that they become resistant to broad spectrum herbicides, such as incorporating selectively into the plant itself rather than the relying on the selectivity of the chemical. Two strategies to engineer herbicide resistance have been adopted. In the first, the target molecule in the cell either is rendered insensitive or it is overproduced. In the second, a pathway that degrades the detoxify the herbicide is introduced into the plant. An example of each strategy is considered here. So, Glyphosate, a non-selective herbicide that inhibits 5-enol pyruvyl syncimate 3-phosphate (EPSP) synthase, a key enzyme in the biosynthesis of aromatic amino acids in plants and bacteria. A glyphosate tolerant Petunia hybridia cell lines obtained from the selection for glyphosate resistance was found to overproduce EPSP synthase as a result of gene amplification. A gene encoding the enzyme was subsequently isolated and introduced into the Petunia plant under the control of CAMV promoter. As I told you, this CAMV is a virus and is isolated from the virus portion. Transgenic plants express increased level of EPSP synthase in their chloroplast and were significantly more tolerant to glyphosate. An alternative approach to glyphosate resistance has been to introduce a gene encoding a mutant EPSP synthase. This mutant enzyme retains its specific activity but has decreased affinity for the herbicide. Transgenic tomato plants expressing this gene under the control of opine promoter were also glyphosate tolerant. Following on from this early research, several companies have introduced glyphosate tolerant into the range of crop species, with soybean and cotton the first to reach the commercialization. Currently, nearly 3/4 of all transgenic plants in the world are resistant to glyphosate. Now next to the term phosphotocretin PPT is an irreversible inhibitor of glutamine synthesis in plants and bacteria. Biolobes produced by Streptomyces consist of PPT and 2-alanine residue. When these residues are removed by pesticidase, the herbicide component PPT is released. To prevent self-inhibition of growth, biophiles produce strains of Streptomyces also produce the enzyme phosphophenol-3-creatine acetyltransferase (PAT), which inactivates PPT by acetylation. The BAR gene that encodes the acetylase has been introduced into potato, tobacco, and tomato cells using agrobacterium-mediated transformation. The resultant plants were resistant to commercially formulation of PPT and bioflaves in the laboratory and in the field. More recently, it has been shown that bioflaves resistant transgenic rice plants which are incubated with the fungi causing seed blight disease and subsequently treated with herbicide were completely protected from infection. This agronomically important result depends on the observation that bioflaves is toxic to fungi as well as by being a herbicide. PPT resistance is widely used in plant as a selectable marker. However, it has been shown to introduce into the number of different crops for weed control, including sugarcane and rice. The benefit of herbicide-resistant transgenic crops is increased yield and seed quality as competing feed species are eliminated. However, there was initial concern that this would come with associated penalty of increased herbicide use, which could have a serious impact on environment. Contrary to these predictions, the introduction of herbicide-resistant plant has actually reduced chemical use up to by 80% in many areas. As farmers adopt better weed control policy and switch to herbicide with low use rate, a further risk is transgene for herbicide tolerant could spread to weed species, resulting in a new breed of superweed. It is likely to say whether this will be a problem. Although a range of herbicide-resistant transgenic plants is being tested, only crop resistant of glyphosate or bromoxylanyl are currently grown on commercially scale. The benefit and risks of herbicide-resistant crops have been revised. Although herbicide resistance is most commonly a strategy for weed control, several others are being evaluated, including enhanced allopathy, in which crop plants are modified to produce their own weed-inhibiting compounds. Typically, the primary crop is the target of modification, but such approach can also be used to produce aggressively cover crops that inhibit weed before the primary crop is planted. There has been recent interest in the use of suicide transgenes inducible by temperature of first period cause the crop to self-destruct at the correct time of year. It is also possible to modify microbial pathogens so the preferentially attack weeds. Now we come to the virus resistance plant. So major crops losses occurs every year as a result of viral infection, such as tobacco mosaic virus cause loss over the 50 million per annum in the tomato industry. There is useful phenomenon known as cross protection, in which infection of a plant with one strain of virus protects against super infection with the second related strain. The mechanism of cross protection is not fully understood, but it is believed that the viral coat protein is important. Now, scientists have developed transgenic plant which express the TMP coat protein, which greatly reduce disease symptoms following viral infection, such that observation principle of heterologous coat protein expression has been extended to many different plants and viruses. In case of resistance of TMP, the coat protein must be expressed in the epidermis and the vascular tissue through which the virus spreads systematically. Transgenic squares containing multiple viral coat protein genes and demonstrating resistance to cucumber mosaic virus, watermelon mosaic virus 2, and zucchini yellow mosaic virus was the first virus-resistant transgenic crop to reach commercial production. A related strategy in which viral replicase was expressed in transgenic rice plant protects those plants from several different isolations of rice yellow mottle virus (RYMV). Because the replicase was more conserved than the coat protein in the best performing line, viral replication was completely blocked over several generations. While the heterologous coat protein approach can be useful, it has been demonstrated that in many cases the effect of transgene expression is mediated at the RNA rather than the protein level. This can be proven by generating transgenic plant carrying coat protein genes that cannot be translated to any functional protein, as was shown by the Dougherty. This transgene RNA apparently interferes with the viral replication. This requires homology between the transgene and the target virus and involve high level of transgene transcription but low level accumulation of transcript features very similar those of the past post-transcriptional gene silencing. Now we come to this fungal pathogen. So progress also has been made in developing resistance to fungal pathogens which are traditionally controlled by appropriate farming practice and application expensive and environmentally harmful fungicides. A straightforward approach is to engineer plants with antifungal protein from heterologous species. This was first demonstrated by Broglie, who showed that expression of bean kinase can protect tobacco and oilseed rape from post-emergent dumpling of or Rhizonia solemnis. Plants synthesize a wide range of so-called pathogenesis-related proteins, PR proteins, such as kinetinase and glucanase, which are induced by microbial infection, and there are now many examples of such proteins expressed in plants to provide protection against fungi. In some cases, Multiple PR proteins have been expressed with synergistic effects in tobacco, carrot, tomato, which have been engineered to express both kinase and glucanase simultaneously. And these plants show greater fungal resistance, indicating that the two enzymes can work synergistically. Plants also synthesize antifungal peptides called defensism, and other antifungal proteins have been shown to confer resistance to pathogen when overexpressed in transgenic plants. Some of these have been shown to provide protection under field trial conditions. As genes from more of these proteins have been cloned and characterized, the number of transgenic plants consecutively expressing such proteins continue to rise. For example, tobacco osmotin has been expressed in transgenic potato providing resistance, and in transgenic rice providing resistance to solanine. Instead of using a protein to provide direct protection, a metabolic engineering strategy can be utilized. Phytoalexins are alkaloids with antifungal activity, and transforming plants with genes encoding the appropriate biosynthetic enzymes can increase their synthesis. Now, some scientists generated tomato plants expressing the grapevine gene for stilbene synthesis, and these plants demonstrated increased resistance to infection by Bortius. The grape stilbene synthase gene has since expressed in tobacco, tomato, barley, rice, and wheat, and has been shown to confer resistance to a range of fungal pathogens. An alternative to the use of antifungal protein or metabolites is to manipulate the hypersensitivity response, which is physiological defense mechanism used to plants to repel attacking pathogens. Resistance occur only in plant carrying a resistance gene that corresponds to an avirulence gene in pathogens. Elictors released by pathogens are detected by plants and activate a range of defense response, including cell death, PR gene expression, phytoalexin synthesis, and deposition of cellulose at the site of invasion, forming a physical barrier. Importantly, a hypersensitive response is systematic so that neighboring cells can preempt pathogen invasion. A recent developed strategy is to transfer a virulence gene from pathogen to the plant under the control of pathogen into insubile promoter. This has been demonstrated in plants of tomato transformed with the AVR9 gene from resulting in resistance to a range of fungal, bacteria, and viral diseases. Now we come to the bacterial pathogens. So, bacterial disease cause significantly loss in crop yield, and many different transgenic strategies have been developed to prevent infection or reduce the severity of symptoms. One of the most prevalent bacterial disease of rice is bacterial blight, which cause losses totaling over about $250 million every year in Asia alone. This disease has been received a great deal of attention due to its discovery of resistance gene complex in a related wild species. The trait was introgressed into cultivated rice line IR24 and was shown to confer resistance to all known isolated of blight pathogens, Xanthomonas oryzae, in India and Philippines. Further investigation of the resistance complex resulted in the isolation of gene named XA21 encoding a receptor tyrosine kinase. The transfer of this gene is to susceptible rice varieties resulted in plant lines showing strong resistance to a range of isolated of pathogens. The XA21 gene has been stacked with two genes for insect resistance to generate a rice line with resistance to bacterial blight and the range of insect plant pests. And this is due to commercial release in China in the near future. As with insect resistance gene, the widespread use of transgenic plant carrying a single resistance factor could promote the evolution of new pathogen strain with counter-adaptive properties. Therefore, other blight-resistant transgenes are being tested for possible development either alone or combination with XA21. For example, rice plants expressing ferredoxin-like protein have been previously been shown to delay the hypersensitive response to the pathogen Pseudomonas stringi in oculation test with all the transgenic plants showed enhanced resistance against the pathogen. So the bacterium Bacillus thuringiensis provides the major source of insect resistance genes. Insect pest resistance one of the most serious biotic constraints to crop production. For example, more than 1/4 of the rice grown in the world is lost to insect pests at the estimated cost of nearly $50 billion. This is despite an annual expenditure of approximately $1.5 billion on insecticide for this crop alone. Insect-resistant plants are therefore desirable not only because of the potential increased yield but also because the need of insecticide is eliminated and following on the form the undesirable accumulation of such chemicals in the environment is avoided. Typically, insecticides are non-selective, so they kill harmless or beneficial insects as well as pests. For these reasons, transgenic plants have been generated expressing toxin and are selectively for particular insect species. Research is being carried out on a wide range of insecticidal proteins from diverse sources. However, all commercially produced insect-resistant transgenic crops express toxin proteins from gram-positive bacteria Bacillus thuringiensis, the Bt. Unlike other Bacillus species, this produce crystals during the sporulation, comprising one or more small number of approximately 130 kilodalton protoxin called crystal proteins. These proteins are potent and highly specific insecticides. They specifically reflect interaction between the crystal protein and receptors in the insect midgut. In such susceptible species, ingested crystals dissolve in the alkaline condition of the gut and the toxin are activated by gut protease. The active toxin binds to receptor on midgut epithelial cell, becomes inserted into the plasma membrane and forms pores that lead to the cell death. Bt toxins have been used as a topical insecticide since 1930s, but never gained widespread use because they are rapidly broken down on exposure to daylight and thus have to be applied several times during a growing season. Additionally, only insects infecting the exposed surface of sprayed plants are killed. These problems have been addressed by the expression of crystal protein in transgenic plants. Bt genes were initially introduced into the tomato and tobacco, and later cotton, resulting in the production of insecticide proteins that protected the plant from insect infestation. However, fields tested for these plants revealed that higher level of toxin in the plant tissue would be required to obtain commercially useful plants. Attempts to increase the expression of toxin gene in plants by the use of different promoters fusion proteins and ladder sequence were not successful. However, examination of bacterial Cry1A, B and Cry1AC genes indicated that differentially significant from plant genes in a number of ways. For example, localized AT-rich region resembling plant introns, potentially plant polyadenylate signal sequences, this ATTTA sequence which can destabilize mRNA and rare plant codons were all found. The elimination of undesirable sequences and modification to bring codon uses into line with the host species resulted in greatly enhanced expression of the insecticidal toxin and strong insect resistance to transgenic plant in field tests. By carrying out such enhancements, Berkeley and colleagues expressed modified Cry3A gene into potato to provide resistance against the Colorado beetle. In 1995, this crop became the first transgenic insect resistance crop to reach commercial production, as this potato marked as new leaf potatoes marketed by Monsanto. The same company also released the first commercial transgenic insect-resistant variety of cotton and maize. Many other biotechnology companies now produce Bt transgenic crop plant resistance to a range of insects. Some of these have been extraordinarily successful. For example, 2 showed the Bt commercially hybrid variety expressing the Cry1AB genes produced a 28% yield increase compared to the wild type plants in the field trials in China. Although Bt transgenic plants currently dominate the market, there are many alternative insecticidal proteins under investigation. 2 types of proteins are being studied, in particular proteins that inhibit digestive enzyme in the insect's gut and lectins. Research into these alternatives is driven in a part by the fact that some insects are not affected by any of the known Bt crystal proteins. Homoplaton insects, mostly sap-sucking pests, are such as plant hoppers, fall in this category but have been shown to susceptible to lectins such as agglutinin, GNA. This lectin has been expressed in many crops including potato, tomato and tobacco. Now we come to the drought resistant plants. After pest and disease unfavorable environmental condition, abiotic stress represents the next major limitation on crop production. One of the most prevalent of these conditions is drought, and make the development of transgenic crops with built-in drought resistance could increase the global yield of food by the 30%. Many plants respond to drought and increase salinity by synthesizing small, very soluble molecules such as betaine, sugars, amino acid and polyamines. These are collectively termed compatible solutes, and they increase the osmotic potential within the plant, therefore preventing water loss in the short term and helping to maintain a normal physiological ion balance in the longer term. Compatible solutes are non-toxic even at high concentrations, so one strategy to provide drought resistance to make such molecules accumulate in transgenic plants. For example, several species have been engineered to produce higher level of glycine betaine, but in most cases the level achieved have fallen short of 5 to 15 micromolar per gram fresh weight observed in plants that naturally accumulating this molecule under salt stress conditions. However, transgenic rice plant expressing BH are likely to be the first commercially released GM plants developed for abiotic stress tolerance and will be available for small scale substance farmers and large producers, as well as compatible solute plants also produce osmoprotectant protein known as dehydrins in response to drought and salinity express. The overexpression of such proteins offers another strategy to generate drought tolerant crop varieties. Yu in 1996 expressed the barley dehydrin HVA1 in transgenic rice, and transgenic plants were shown to be resistant to water defects and salt stress. More recently, several wheat dehydrins have been expressed in transgenic rice and have been also shown to the increased dehydration tolerance. Now we come to the poor soil quality. About 65% of the world potential uploaded land consisting of marginal soils, which are characterized by extreme pH, limited nutrition availability, and high level of toxic metals and acidic soils, which accumulate for 40% of arable land, have low level of available phosphorus and iron but high level of aluminum. In an acidic environment, both iron and aluminum filter phosphorus into the insoluble or poorly soluble molecules. Aluminum is also toxic in its own right, its major effect being the inhibition of root development. Alkaline soil accounts for 25% of arable land. The major problem of alkaline soil is to high level of calcium and magnesium ion, which also sequesters phosphorus into the insoluble and sparing soluble molecule. Calcium is an essential signaling molecule in plants, and high level of this metal ion can interfere with the normal plant growth and metabolism. Despite these problems, many plants have adapted to grow in marginal soils, and some tolerant variety of crop plants have also been produced by mutation and conventional breeding. A common factor among these tolerant plants is increased exudation of organic acids such as citrate, malate, oxalate from the root. The production of transgenic plants ingredients to exude high level of organic acids is therefore an alternative strategy to increase the use of marginal soils. Several species have been transformed with the bacterial or plant crystal synthesis gene to increase organic acid production and induce tolerance of poor soil. The analysis of root extract by HPLC showed that citrate levels were 10 times higher than normal, but more importantly, the level of citrate recovered in root exudate were 4 times as the normal level. These plants could grow in presence of aluminum ions at 10 times the concentration sufficient to suppress the growth of non-transformed plants, and there was no evidence of aluminum inducing root damage. The transgenic plant also performed better in alkaline soil. They grew and flowered normally and produced seeds, whereas control plants showed restricted vegetative growth and failed to produce flower and seeds. The exudation of phytase from the root of transgenic plants can be used to release phosphorus locked away in organic compounds. Since much of the organic phosphorus in soil is presented as phosphatase, initially experiments with Arabidopsis plants secreting phytase from the root showed that the increased transgenic plants were able to grow much better than control plants on the phytase medium. The development of crop plants of secrete phytase and organic acids is therefore an important goal for the future. coming to the last topic, that most important goal of plant biotechnology to increase food, the amount of usable food obtained from a field of plant can be tremendously. As much effort has been expended in attempts to increase yield by conventional breeding and optimization farming practices. Genetic engineering provides a wide range of strategy not only to reducing yield loss but also by increasing the intrinsic yield potential of the plant. In terms of yield enhancement, photosynthesis is perhaps the most obvious target of genetic intervention because all determines the rate of carbon fixation and therefore the overall size of the organic carbon pool. Strategy to increase photosynthetic activity include multiplication of light harvesting photochromes and key photosynthetic enzymes. Progress has been made in crop species by attempting to introduce components of the energy efficient C4 photosynthetic pathway into the C3 plants, which lose a proportion of their fixed carbon through photorespiration. The key step in C4 photosynthesis is conversion of CO2 into C4 organic acid by enzyme phosphoenol pyruvate carboxylase (PEPC) in mesophyll cells. The maize gene encoding PEPC has been transferred into the several C3 crops including potato and rice in order to increase the overall level of carbon fixation. Transgenic rice plants were also produced expressing pyruvate orthophosphate dikinase and NADP male enzyme. Primarily, field trials in China and Korea demonstrated 10 to 30% and 30 to 35% yield increase of PEPC and PCDK transgenic rice plants, respectively, which was quite unexpected since only one C4 enzyme was expressed in each case. Metabolic approaches to increase overall yield focus on the conversion of sugar representing the direct production of photosynthesis to the bioavailable storage carbohydrate starch. Several different studies have been attempted, including the manipulation of enzyme activity and regulation in source tissue to increase the carbon flux to sink tissue, increasing efficiency of sugar transport to sink tissue, and the manipulation of enzyme activity is the sink tissue itself increase the conversion of photoassimilation into the starch. Transgenic approach to increase sugar to starch conversion have focused primarily on the manipulation of plastidyl starch synthesis pathway. For example, inhibition of plastid adenylate kinase in transgenic potato resulted in a substantial increase in the level of adenylase and the 60% increase in the level of starch in combination with 40% increase in tuber yield. Other strategies that have been used to include the modification of plant architecture, diverse resources to harvestable products in rice, manipulation of the gibberellin signaling pathway by overexpression of mutant arabidopsis GAI gene was sufficient to induce a domain semi-darwinism phenotype. Developmental genes have been expressed in an attempt to increase the number of spikelets in maize, while other researchers have attempted to modify the way plants respond to day length or seasonal clues to induce early flowering and increase number of harvestable crops per year. So up to here we have discussed everything which is the application of biotechnology in plants. So thanks for listening. Now this is finished.